Hi students, welcome to HP Carpenter School. I am Nisha James and I am taking biology practical. Today we are going to do another 12th standard biology practical, that is to verify Mendel's law of independent assortment. So if in examination, if this question is asking to verify Mendel's law of assortment, what you have to do? You have to do dihybrid experiment. This is very important question. This question was asked in last year board examination. So now I am going to tell you how to perform that practical. Okay, so I am starting the practical. The practical number 14, to verify Mendel's law of independent assortment. So how we can verify Mendel's law of independent assortment? This can be verified by dihybrid experiment. So what is a dihybrid experiment? An experiment at a time which is focusing on two characters, that is called dihybrid experiment. For example, the character selected by Mendel. Okay, or the character at a time we have to take two characters. So what are the two characters we are taking? The first one is the color of the seed and the second one is shape of the seed. And the plant that we are taking for experiment is Pisum sativum or garden pea. So when you observe garden pea plant, they have the seed is occurring in two colors. Okay, what are the colors? Some of the seeds are yellow color and some of the seeds are green color. And shape of the seed is also variable. You can find round type of seed and also wrinkled type of seed. The round type of seed is a dominant character because it is shown by most of the individuals of the pea population. And yellow color seed also a dominant character because it is also observed by most of the pea plant population. So now I am going to tell you how to do the practical. So the characters we have selected is shape of the seed. I told you that the shape of the seed, there are two types, round seed and wrinkled seed. Round seed is represented as capital R, capital R. Wrinkled seed is represented as small r, small r. Why this capital R, capital R? Because as I already told you that round seed is a dominant character. The alphabet of the dominant character always represented in capital letter, and that of the recessive character, that is represented in small letter. Now, what about the color of the seed? That is our second character. That is yellow and green. Yellow, as it is a dominant character, it has to be represented in capital letter. So that's why we write capital Y, capital Y. And the green color, that is a recessive character, so I have represented with small y, small y. And then you can find that, okay, so these are the two characters we are taking for the experiment. When the genes are, how the genes are segregating, what are the chances of various combination, that we will explain, that I will explain now. Okay, so first we have to produce F1 generation. How to produce the F1 generation? First we have to take a male plant. What about the characters of male plant? Male plant is round and yellow. Then it is crossed with a female plant. What about the character? That is wrinkled and green. And then what about its genotype? The male plant genotype, as it is round yellow, it is represented as capital R, capital R, capital Y, capital Y. The female plant character, as it is a recessive word, it is represented as small r, small r, small y, small y. As I already told you that in monohybrid experiment, each character is represented by a pair of LED. That's why for yellow color, capital Y, capital Y. And then for round seed, capital R, capital R. Okay, so I think all of you understood that. Now, what about the breeding? Now, during the time of gamete formation, the capital R is split into R and R. Capital Y will split into Y and Y. The same thing is happening in the female plant. The small r, that is a wrinkle, that is split into R, small r and small r. And the small y, that will split into small y and small y. So these are the gamete. You can find that the gamete that has to be encircled. So each of the gamete is having a single set of a single character. Okay, each of the gamete is having a single character. For example, double R, now it is single R. When you observe this capital Y, each of the gamete will get only one Y. So this is applicable to the female plant also. Okay, so as this is a homozygous one, capital R is a homozygous because both the alleles are same. What is the meaning of homo? Homo means similar. When the gamete consists of same allele, it is called homozygous. So as it is a homozygous one, I am taking only capital R, that is representing the round seed, and from the capital Y, I am taking only one Y because it is also homozygous. So the gametic character we are getting is, the dihybrid gametic character we are getting is capital R, capital Y. Gamete consists of two characters. First one, the shape of the seed, and the second one, that is capital Y, that is the color of the seed. Now, when you go to the female plant, what about the character? The character you can find, this is also homozygous. So from this capital R, we have to take only small r. Okay, from this small r, we have to take only small r. From the small y, we have to take only small y. So, what about the gametic character? It is small r, small y. This is also representing a single character of each. Small r is representing what? Wrinkled seed. Small y is representing green color seed. Now, there will be fusion of male gamete. What is the male gamete? Capital R, capital Y. And what is the female gamete? Small r, small y. After this fusion, that is fertilization. What is fertilization? Fusion of the male and female gamete. We will get F1 plant. Now look at the anatomy of the F1 plant, genotype of the F1 plant. F1 plant is capital R, small r. So I am writing capital R, small r. Now capital Y, small y. So this is the genotype of the F1 plant. So what is the genotype of F1 plant? Capital R, small r, capital Y, small y. So we got the genotype. From the genotype we can conclude what about the phenotype. As you know that whenever a heterozygous allele is there, okay, then only dominant character will express. So capital R, small r is a heterozygous. So dominant character क्या है? Capital R है. So that the round character will capital R is round. So that character will express. So we are getting round. Capital Y, small y is again a heterozygous. So the dominant character will express. What is the dominant character? That is yellow. So what is the phenotype? The phenotype of F1 plants were round yellow. So in this way we have to calculate the F1 generation. Now we have to calculate F2 generation. So how to calculate F2 generation? For F2 generation we have to take two F1 plants. So these are the two F1 plants. One is a male plant and another one is a female plant. The male plant, what is the character? Capital R, small r, capital Y, small y. Okay, the same character that we have taken here. Capital R, small r, capital Y, small y. This is a male plant. This is crossed with the female plant. What is the female plant? That is capital R, small r, capital Y, small y. Now what will happen? During the time of gamete formation, the pair will segregate into individual one. Okay, so the gametes will separate. This will separate into capital R, small r in this way. This will separate. Capital Y, small y in this way it will separate. Okay, now I am writing the gametes. Every time you have to remember gametes should have both the character, that is the color of the seed and shape of the seed. So to represent the gametic character I am using a Punnett square. Because here the number of gametes are more. When the number of gametes are more, we have to use Punnett square. So this is a Punnett square. Then the vertical columns and horizontal rows, that is mainly depending on the number of gametes. So in the top of the Punnett square, I am representing the male gamete. What are the male gametes we are getting from this? That is capital R, capital Y. Then capital R, small y. Then small r, capital Y. And also small r, small y. Okay. So how many vertical columns? 1, 2, 3, 4. Now what about the horizontal rows? You can find on the side of this Punnett square we have to represent female character. So this is the female representation. What about the gamete? Same as that of the male gamete because characters are same. Capital R, capital Y, that is the first gamete. Second gamete is capital R, small y. Third gamete is small r, capital Y. And the fourth gamete is small r, small y. Now we have to do hybridization. Okay, so we have written male gamete on the top of the Punnett square and female gamete on the side of the Punnett square. The gamete has to be encircled, that you have to remember. Now how to do the hybridization? Okay, the male and female gamete will hybrid. It will undergo fusion. So what to do that? First you have to take one of the male gamete, allow it to fuse with the 4 female gamete. Then you have to take the second male gamete, allow it to fuse with the 4 female gamete. In this way, all the 4 male gametes fused with the female gamete and we are getting the 16 combination, 16 genotypic combination. So I will tell you one or two examples. Okay, so first I am taking capital R, capital Y and allowing it to fuse with the female gamete. So this one I am taking first male gamete and first female gamete. So what we will get? Capital R, capital R. So I have written capital R, capital R, capital Y, capital Y. Now I am taking the male gamete with second female gamete. So what we will get? Capital R, capital R. We'll go capital R, capital R and capital Y, small y. Now we have to take the third one. Okay, so this first male gamete and third. So what we will get? Male gamete का है capital R, female gamete का है small r. So capital R, small r. And then male gamete capital Y, female gamete also the capital Y. So what will happen? Capital Y, capital Y. Now the fourth fusion I am telling, the male gamete capital R, capital Y, female gamete small r, small y. So what will be the genotype? Capital R, small r, capital Y, small y. So in this way you have to do the crossing. So take one of the male gamete, allow it to fuse with the female gamete. In this way you have to flip all, you have to do crossing of all the male gamete with the female gamete. Then we will get 16 genotypic combination. I have written the genotypic combination. Okay, so you can count it 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So 16 genotypic combination we are getting. Now we have to analyze what about the phenotype. Genotype तो हमको मिल गया, अभी phenotype कैसा है? So that you have to write in short form on the top of each. Capital R, capital R, capital Y, capital Y. Both the characters are dominant, so it will be round yellow. Capital R, capital Y, small y, that is round yellow. Again, that is a dominant characteristic. So in this way, you have to fill the phenotype of each of the particular column. Okay, so I have written the phenotype. Now, after completing this, what you have to do? What is the next step? You have to write the ratio. So first we will write phenotypic ratio. Phenotypic ratio we will get, this is the phenotypic ratio. Okay, what is the phenotypic ratio? That is after counting this number, okay, phenotypic character is round yellow. So how many round yellow we are having? We have to count 1, 2, 3, 4, 5, 6, 7, 8, 9. So I will say we have 9 round yellow. Now what is the second combination we got? That is a round green combination. That is a new combination. It is different from that of the parental combination. So we have to count how many round green we are getting. Round green can be represented as RG. So I am counting how many round green? 1. Okay. Now 2 and then 3. So how many round green we are having? Round green is 3. Now the third combination, phenotypic character is wrinkled yellow. So how many wrinkled yellow we are having? Wrinkled yellow hai, we have to take 1, 2, 3. So I have written 3. Now what is the last combination? That is a wrinkled green combination. So how So what is the phenotypic ratio? That is 9:3:3:1. So in a diagram experiment, we are getting a phenotypic ratio if 9:3:3:1. Here we are getting two parental characters. What are the parental characters? Round yellow, that is 9, and the wrinkled green, that is 1. In between two new combination we are getting, that is round green and also wrinkled yellow. Now what about the genotypic ratio? The genotypic ratio of F2 generation is 1:2:1, 2:4:2, 1:2:1. Once again, what is the ratio? 1:2:1, 2:4:2, and 1:2:1. When you do the totaling of all this, you will get 16. So by this, okay, we have verified. So we are getting this 9:3:3:1 phenotypic ratio and the genotypic ratio also. So by analyzing this data, we came to a conclusion that the segregation of one gene, that is independent of another gene. That's why we are getting new combination which is different from parental character. I told you that parental character ke alawa we are getting two new combination, that is the round green and also wrinkled yellow. So this is verifying the Mendel's law of independent assortment. So this is very important question. This question was asked in last year board examination. This time also we can expect this. So this is the practical. Thank you.